Ya.
Vale, creo que ya estamos en directo. Bueno, bienvenidos a otra edición de este BioInfoClub. En primer lugar, informaros, como se ha dicho por redes sociales y todo, que esta charla y las preguntas y todo será en inglés. So, welcome to another edition of this BioInfoClub. This talk and the questions and everything will be in English, as we already said in our social media. So, we hope you enjoy it. My name is Anne, and I'm pleased to present you this activity. So I'm presenting today the PhD student Pablo Iañez. He did his master's in bioinformatics in the University of Copenhagen, and now he is a research assistant in the Cellular System Genomic Lab at Josep Carreras Leukaemia Research Institute in Badalona, Spain. And he is interested in the application of machine learning and deep learning models on biodata. So today he will explain us on how to work with generative adversarial networks that are coupled with the Markov Chain Monte Carlo algorithm. So it's quite interesting, and I think it's a topic like really technical, but I think we're going to enjoy it. So, Pablo, the floor is yours.
Okay, thank you so much, Anne Martinez. So, first of all, yeah, thank you, Anne, for inviting me to these talks, and especially thanks to everyone who is behind BioInfoGRX, because I think that they are doing an amazing job. I think it's a great initiative to promote the bioinformatics at national level, but especially in a city like Granada, with so many talks and webinars and courses and all. So as Anne said, I'm going to present today the project that I did during my master thesis in the University of Copenhagen. This was done in the Geogenetics Center and Globe Institute in the group of Fernando Racimo. My advisors in that time were Graham Gower and Fernando Racimo, and the title is Demographic Inference using Markov Chain Monte Carlo coupled Generative Adversarial Networks. So this is the structure that I'm going to follow, the typical structure. I'm going to start with an introduction of what are the bricks that I'm going to use in this architecture that I'm going to show. Then I'm going to move on to the materials and the methods. I'm gonna speak more calmly about each of the different part of this super big model. And then results and discussion, conclusion and bibliography. So in order to start with the introduction, in the field of population genomics have been a lot of recent advances and progresses in the last few years, mainly thanks to the advances in the sequencing technologies. So now there are a lot of consortia and big projects such as the 1000 genome projects that aim to sequence and characterize the genotype of like massively a big number of individuals. In order to give an understanding and to study this, this observed genomic data, it is usually encapsulated in a mathematical theory, in a mathematical framework that is called the coalescent theory, which is just a stochastic evolutionary model of gene evolution within a given population. So in order to estimate these demographic models in this framework, one could compute the likelihood of the data given a demographic model, but this is sometimes not feasible because this works analytically in small models, but when the model is bigger, then you cannot really do this. It's very hard. So one solution that has been studied a lot, there are a lot of papers about it, is to use likelihood-free methods, such as the approximate Bayesian computation. I'm not gonna enter into details on this, but it is affected by one concept, that is the curse of dimensionality, in which as the number of dimensions increase in your dataset, then you have also an increase in the neighboring empty spaces around it. And basically, the computational times and the complexity increases exponentially. And also, using this method, you need to compress the whole genomic data into summary statistics, so there is a loss of information. More recently, deep learning methods have been applied almost in everywhere that you can apply this. It has been a buzzword, but it has been proven to be very, very efficient on the population genomics field. So deep learning are a subset of machine learning algorithms that permit to work, in this case, with a complete genomic data by learning a useful representation of it. So more specifically, I'm going to talk about the convolutional neural networks. That is the one that I'm going to use in my architecture. So this is the standard architecture of a convolutional neural network. Looks like this. We have the input images. There are different layers. The more layer you have, the more deep is your model. The first part consists on the feature extraction. So you can see that the image slowly gets smaller, but at the same time, we have a higher number of these images. First, you have a convolutional operation, and then a function to aggregate the information in a more condensed space, doing the downsampling of your data. That is called pooling. There have been like max pooling, average pooling. There are different functions that you can use. And then in the end, you end up with a vector that you can give to the output layer. And here you can do the regression or classification depending on the problem that you have at hand. So here I have a depiction of what is a convolutional kernel. Usually you have tens and hundreds of these in your convolutional layer. And what it does is basically it tries to scan the input image to see if it's able to extract, to identify the pattern that it has learned during the training. So the learning of this architecture is an optimization model, optimization problem that is solved with a class of algorithm called backpropagation, meaning that once you go to the output, you perform gradient descent backward in the network in order to recalculate or reconfigure these numbers of your matrices in your model, so the weights of your model. This is just one part of the bigger model here that is called the Generative Adversarial Network. I'm just going to call it GAN. I think it's easier. So they are fully probabilistic model of the observed data. The goal of them is to synthesize fake samples that are realistic as the ones coming from the real data that you give to the model. So here you have two neural networks, the discriminator D and the generator G, and they both compete one against each other in a zero-sum game. The goal of the generator is to simulate fake data that mimics the data distribution learned from the real samples. And at the same time, the discriminator goal is to avoid being fooled by the generator fake samples and to be able to differentiate or to discriminate between the real and the fake samples. So this is what I said, but just put it in a diagram, in more in context. Having a lot of have a lot of applications, but here just for fun and for the simple example, I use a picture from a Mr. Bean movie, but it can be, for example, photos of dogs or cats or whatever you want to learn, faces, for example. And this is a batch of image that we consider the real data, which have a label of 1. At the same time, we have a series of fake samples that have been synthesized by the generator, and they create the fake samples. So y equals to 0. They have the label of 0. These two batches of pictures are given to the discriminator, which is going to try to infer which one is real and which one is fake. So basically just output a prediction, a probability. If the probability is 1, it means that it's super sure that it comes from the real samples. If the probability is 0, it means that it's sure that it's coming from the fake samples. So ideally, after the training of this architecture, you would end up with a discriminator that output a prediction of 50%, meaning that he's not sure from which source it's coming from, and he's just guessing randomly. So one that you are training this, and you have the prediction output, you give it to a loss function that is later used to reconfigure, as I said, the weights both in the discriminator network, but also in the generator network. So in each training iteration, they both get better at their own individual goals. They have been applied already in some cases. I think they are from 2015, so they are quite new, but they have been already used to create realistic, photorealistic images, which are called deepfakes. Actually, they are a big problem right now because they have been used a lot in social networks like Facebook in order to create scam and to fool people. And I think like some governments are already implementing rules and some new laws in order to control these deepfakes. It has been also used to generate and learn 3D objects and even video data, which I think you can find on Google, and it's quite amazing, the video that you get from it. But also more specifically in the bioinformatics field, Maruf et al. In 2018, they used it to recreate realistic single-cell RNA sequencing data. Gelman and colleagues on 2019 used it to simulate artificial human genomes that they claim that can be used to augment the available public data. And then more recently, in 2020, Wang et al. Did it for automatic inference of demographic parameters. So super similar to what I'm going to show today. But instead of Markov Chain Monte Carlo, they used another approach called simulated annealing. So I cannot employ a standard GAN architecture for this case. And this is because normally in population genetics, you would use a simulation engine in order to create new data samples, so like a new variant data. This simulation engine can be a library or a package that you import into your script. But these applications, of course, they are not differentiable. They are not neural networks, and therefore they cannot be adjusted with the standard backpropagation algorithms. So they act as a sort of a black box. So you give the input, you have the output from the simulator, but you cannot configure with backpropagation. So there have been several proposals like the simulated annealing from Wang, as I mentioned before. And here we're going to focus on another alternative that is called Markov Chain Monte Carlo. So just to give a clear idea of after the introduction, the problem, as I said, is that we want to work with the whole genomic data in order to learn the parameters for a demographic model, but also that different evolutionary models and a combination of values of parameters can lead and explain the same observed data distribution. So in our approach, what we want to do is to develop this MCMC GAN in order to discover the joint probability distribution of the demographic parameters. In our case, the discriminator is a permutation invariant neural network, and the generator is a Markov Chain Monte Carlo coupled MS prime, which is the software that we use to simulate variant data from the coalescent simulator. The advantages is that it allows for automatic inference of the parameters from the demographic models, that at the same time, you can generate realistic population-specific genetic data that mimics real data sets. You can also augment this, use this to augment the public databases. And also it works with the whole distribution of parameters, not the maximum a posteriori. Maximum a posteriori is just one number, one scalar that explain or collect your distribution. But of course, there is a lot of information. Jumping onto the materials that I've used, the main libraries that I have used in my code, which it was all programmed in Python. It's MS Prime, which is a library that is used to simulate coalescent scenarios under different evolutionary models. This is quite flexible, so you can give different values to different specification of your model, and you get the variant data from it. Standard PopSim, which is uses MS Prime as the backend, and it also simulates variant data using coalescent scenarios, but with published demographic models from the literature. Then I use SAR for the compression, storage, and processing of big matrices. And I really recommend this library. Have been a good pleasure to work with it. And then for the deep learning part, I use TensorFlow, which is a machine learning platform with an ecosystem of adds-on like TensorFlow Probability, and Carreras, that is a library that is built on top of TensorFlow. It's a higher level and is used to build deep learning models. The data comes from, well, both simulation, but the real data comes from variant call files from the 1000 Genomes project, in which I have applied a genomic mask to filter out bad sequencing quality or regions that are uncallable. And for computing platforms, I have used the GPUs and TPUs from Google Colab, but later on I moved to the servers from the Geogenetics Institute. Then the methods. This is how the modified GAN would look like. So I'm not giving pictures of any painting or dogs or any animal. The picture that I'm giving are actually matrices that are called genotype matrices that I'm going to explain later on. Both the real samples and the fake samples are given to the discriminator, which is a permutation invariant network. And using just the D-score, so the prediction of the discriminator, is given to the Markov Chain Monte Carlo in order to gather a new batch of proposed values for the demographic parameters, and then given to MS prime, which would act as the generator in this architecture. I'm going to talk step by step, part by part of this about this architecture. So I'm going to jump into the data, the genotype matrices. Each data point is this kind of matrices that are composed of values of only zeros and one, and have a dimension of h times s, where h is the number of haplotypes or rows. So here, for example, this is an example. I have 5 rows and 5 haplotypes. We humans are diploid, but just to reduce the dimensionality, I have used 1 haplotype for each individual. And then S is the number of segregating sites or colon. So it's just the number of places in which we are known to have SNPs in our genome. Then at each specific index, the value is 0 if it belongs to the major allele, and it's 1 if it belongs to the minor allele. A part of these dimensions, I also include the batch dimension, which is just the number of replicates that I have, and the channel dimension. The channel dimension for color photos, for example, you would have 3 channels corresponding to the red, blue, and green. But in my case, I just have zeros and ones, so 1 is enough. So for 100 replicates, I end up with a tensor of 100 replicates, 5 haplotypes, 10 SNP sites, and 1 channel. Before feeding it to the discriminator, I do some pre-processing for each matrix. First, I look for the samples, sample identifiers that belong to the desired population, for example, the Utah resident with European ancestry. So I make sure that I only work with individuals from this population. Then I extract the data from the variant call files, and I pre-process it with SAR, and I store it on disk. Then I sample the genomic region randomly. Some chromosomes are bigger than others, so the sampling is done having this into consideration. From this region, I extract all the biallelic SNPs within 1000 kilobase pairs after filtering by the genomic mask, and then I polarize the matrix by major and minor allele frequency, meaning that I just put zeros and ones. I also filter by the minor allele frequency using a threshold of 0.05, meaning that those alleles that are very rare in this population, I'll filter because they are not giving us any meaningful information. And then the SNPs dimension is just resized to a fixed size of 128. So it makes much more comfortable and faster for all the calculations. For the chromosome 12, from this base pair to this base pair, the genotype matrix would look like something like this. And then I reiterate that the empirical data would be like the class with the label y equals to 1, and then the simulated data from MS prime is the class with the label y equals to 0. Then the discriminator, it's a permutation invariant network. have been also quite fashionable in the field of population genomics. It have been used in several papers already. The motivation is that, as I showed it before, in the kernel, in the convolutional kernel, it was two-dimensional, so it was like 3 per 3. But in our case, we don't really have any pattern as an ear of a dog or something that we want to identify in 2 dimensions, but rather we would prefer to work in the haplotype, in only one haplotype at a time. Also because the haplotypes from a population should be exchangeable, meaning that if I exchange the haplotype from one individual by another one, this shouldn't create any difference to the prediction of the discriminator, because it should be explaining the same data distribution, right? Because they are coming from the same population. So how we do this is that we've applied kernels, convolutional kernels that are only 1 dimension. So for example, this one have 1 dimension and length 4. This is used to scan over each haplotype independently. And then because I use a stride, meaning that I, instead of jumping in a window of 1 by 1, I skip it in 2, so in 2 in 2. Then I cut the length by half, and I end up with a matrix that it's 3 rows and 6 columns. But we also want to combine the information belonging to different individuals. And we do this by applying a symmetric function. The symmetric function can be any, like the maximum, the minimum, the sum, the average, but it is done on each row in the same way. So we end up with a vector of the length 6. This is a representation of how would be a discriminator in my architecture. So you have one input that is a genotype matrix. You give it to the convolutional layer that uses an activation function that is called leaky rectified linear unit, and I also normalize for each batch. Then you would apply asymmetric functions to collapse the dimension and end up with this 2D matrix, and then another convolution, and then another symmetric function. So by repeating these 2 times, you end up with a vector of length 64 in this case, which goes later to another dense layer and finally to the output layer. Because this is a binary classification, whether I just want to predict if yes or no, then I'm using cross entropy as the loss function. And this network is inspired in the all convolutional neural networks, meaning that there is no pooling layer, it's only convolutions. And finally, the Markov Chain Monte Carlo. I'm going to try to explain it with the simplest way that I can. A Markov chain, it's a model that tells you what is the probability of jumping from one stage to another one, like from a sunny day to a cloudy day. And then Monte Carlo methods are numerical methods that are used to estimate, to approximate any quantity of interest just by drawing a lot of random samples from it. And together they merge and they create the Markov Chain Monte Carlo. So this is a series of methods that are used to draw a sequence of correlated samples that will eventually converge to a target distribution that you don't know and you want to infer. So here you are proposed a new state x prime given the previous state x from the proposal distribution using this formula. And this in the Metropolis Hastings algorithm, which is one of the easiest and most simple Markov Chain Monte Carlo, the new state x prime is accepted using the acceptance probability alpha, which I'm not going to enter into details, but it's used to guide the MCMC chain toward regions that are more likely in the target distribution. I know that this is a mess, but I hope that this representation explains it better. Just to mention that the kernel that I use in my case is called HMC, or Hamiltonian Monte Carlo, which is a very sophisticated Markov chain kernel that uses Hamiltonian dynamics to detect the topology of the distribution. So if you imagine that we have this distribution this is the parameter space. So we have two parameters, this one and this one, with different values. And we see that the unnormalized probability distribution is a bimodal distribution where the most likely areas with highest probability is here and here. If you compute the energy function of this distribution, you end up with this topology that it looks like a skate park. And Markov chain is just this chain, this red chain that you see here. It looks like a skate boy bouncing off the walls on the skate park. So it's just a chain that is able to detect and move accordingly to this topology. It departs from one state, which is, I don't have time to point it, but it's the point from which it starts, and then the point in which it ends is the proposed new state, which can be accepted or rejected. And the blue dots that you see here are all the new states that have been accepted. So if you run the simulation and you take, I don't know, 1 million states, then you would end up approximating this unnormalized probability density, because most of your blue dots will fall in here, in this parameter space, or here, giving space to this bimodal distribution. The advantages of this Hamiltonian Monte Carlo is that you have more successful proposals, which translates to fewer sampling steps, which translates at the same time to faster and higher accuracy. And now, just to finalize this part, I'm gonna look at the whole picture as a whole and tell you how we do one single training iteration. So first, we gather data both from the real class with the label 1 and the fake class from the label 0, which are generated, shuffled, and split 9 and 1 into the training dataset and the validation dataset. Then I compile and train one neural network as a discriminator D, and the target that I want to infer using the Markov Chain Monte Carlo is going to be the log density distribution of the scores of the discriminator. So here, the Markov Chain Monte Carlo is going to propose a new combination of parameter theta prime that is going to be given to MS prime, and I'm going to generate new genotype matrices, which should be updated and more closely related to the one from the real data. Then the acceptance probability is going to be computed only using the average of the scores, in order to know from the discriminator if this new combination is actually closer to the real data or it's just bullshit and we can discard it. And then once that I have accepted, then the new samples that I have collected conforms the distribution of the generator. I give it to MS prime, and it's going to generate new genotype matrices with the updated batch of demographic parameters. And the discriminator is going to be, again, trained in this new real and fake data. So the fake data is supposedly more realistic, so the discriminator should be more challenging for him to differentiate between the two sources. Now I'm going to jump into the results and discussion. First, I want to show just the discriminator inference power. Here, we are not looking at the whole GAN. This is just a discriminator. I want to show you what is the distribution that we are giving to the Markov Chain Monte Carlo, so to the skateboy. The discriminator is able to learn the distribution in an effective and deterministic manner. So it's guiding the MCMC, which is much more stochastic, towards the real distribution of the data. In order to do the experiment, I gave two different data sets. So the data set A, or the real one, has a fixed parameter value that I want to infer. In this case, it's going to be the recombination rate, or R, that is a very small number, in this case in logarithm scale, but this is 10 to the power of -8. Is this vertical line, the real value that I want to infer. And the data set V that contains random parameters values that are collected from a uniform distribution. So I collect values from 10 to the power of -10 to a higher one, like 10 to the power of -7. So for each training iteration, which is called epoch, you can see that in the beginning, there is not a real or a clear distribution that can be given to the Markov Chain Monte Carlo. I can give this distribution, but it's not going to be able to do any accurate prediction. And as the training goes on, and I reach 50 different training iterations, it creates this Gaussian distribution that have the mode, the parameter that I want to infer. Here we can also see on the left that both the training and the validation accuracy for this network over the 50 epochs of training. And another example, instead of the recombination rates, here is the effective population size, which the vertical line signals that is 10,000, the one coming from the real data. And then we can see again that over the epochs, this time it's not a very clear Gaussian distribution, but at least you can see that it gets like a unimodal distribution, that the mode is again the parameter, the value that is the real one. So if we only want to infer a single parameter, it's much more easier than inferring the joint probability of different one at the same time. But here I just want to show you how the collection of new parameters values gets more solid as we move on in the training. So here I get 1,000 samples in each training iteration for the Markov Chain Monte Carlo. It takes 50 minutes per training iteration, so it was quite a lot, but I have done some time optimization afterwards. And the real value, it was 10 to the power of -9 for the recombination rate. And the estimate that I get is 1 to the power of -8.97, which is quite accurate with a standard deviation of 0.14 and a convergence after 9 iterations. So you can see in the beginning, the distribution, it's just spread all over different values. And then the more we train the whole GAN, here it already shows more or less a good estimation, a good distribution, but somehow in the next iteration, it gets a little bit tilted to the left. But again, after the 9th one, we end up with this distribution. This, as I said, gets increasingly complicated the more parameters we add to our model. So here is another very simple model. It's a demographic model in which we have a population that had a stable effective size in 1. Then at time 1, suddenly it expanded to the population size of N2. And then at time T2, it had an exponential growth until the present day. So this would be the size at the present day. The real values that I give to it is T1 should be 500 generations, T2 3,000 generations, and the growth rate 0.01. Here I depict both the marginal probability for each one of these parameters. This will be for T1, this is for T2, this is for the growth parameter. We can see that the growth is the one that is inferred with the most accuracy, while T1, it was hardest for the model. But once we come up with this topology map in which we mix, we do a pairwise comparison of each of these parameters, we see that the mode, the estimation, and also the distribution that the two-dimensional distribution that we end up, it's quite accurate with the fixed parameter that I wanted to infer, which is the red dot over here. And this is just a pairwise comparison 2 by 2, but you should think of this as a distribution in 3 dimensions because we are working with 3 different parameters. This is quite a simple model. So imagine working with one of 20 different parameters in which we end up with a density existing in 20 different dimensions. So this is an ongoing project, still haven't been published, have been having some trouble getting the GAN to a stable equilibrium and a stable training because the GAN are already infamous for being really hard to train. And here that we modify using the Markov Chain Monte Carlo, that is a very highly stochastic component, is even harder. But it's already on the way and very, very near a publication. So jumping to the conclusion and future prospects, not just conclusions, actually. We have shown that a prototype of MCMC GAN that is able to infer demographic parameters from segmented data. we have shown that the permutation invariant discriminator is actually good working with quite small data sets. It improved a lot the training times. We have seen that the prediction uncertainty from the discriminator can be used to jointly infer fixed parameter values, but some parameters are harder to infer than others. So this can be quite problematic if we work with 10 parameters and 3 of them are problematic. Then we have seen that the Hamiltonian Monte Carlo sampling is an effective proposal distribution in a generative adversarial framework. And if we couple this with a software like MS Prime, it's a good alternative to a normal convolutional neural network as the generator. And also I want to point out that TensorFlow documentation have been really hard to understand. And there are scarce examples on the internet on how to proceed. So actually, if I could start from scratch, I think I would just use a different library like PyTorch. And finally, I want to give big acknowledgements to people who have been super important and have been guiding me a lot during this project. This project hasn't been easy at all. I have learned a lot. It has been fun, but also very challenging. And this was done during the pandemic times, so we were all working from home. But I need to thank Graham Gower, which have a lot of patience with me and have been a big inspiration. And also Fernando Racimo for his very insightful suggestions. And of course, all the members of the Racimo group in general. So that's everything. And I guess now it's question time.
Thank you, Pablo. It was really interesting, really, I don't know how to say it, like really easy to follow. And actually, I don't know nothing about deep learning, so I actually learned something today. So that's good. So, so questions. I have also some questions for me. So, first of all, they asked us if there is any way, or if you want, you can stop sharing.
Okay.
So first of all, they ask us if there is any easy way to adapt your model and train it for other specific demographic models.
Yeah, so we have been working and testing and debugging this with a few models with increasing complexity. We have tried to come up with a discriminator that is good in general in any model that you give it to it. But we have seen that the architecture of it is determinant to how well it works on each model. So maybe for demographic models that are bigger with more parameters, maybe your discriminator should be also bigger and more complex, maybe by adding more kernels or more extra layers. But yeah, we would like it that it's universal, but it looks like it should be adapted depending on each case.
Okay, thank you. Then I have more a personal question.
Yeah.
In my master's, we learned about Carreras and TensorFlow. We didn't go deep on it, but could you tell us a little bit, or I wonder, which is your experience with it, or what's your personal opinion about these two Python packages that are like super known for doing deep learning?
Yeah. So I started with Carreras. I think everyone starts with Carreras because then you see how you can build a model in just 5-6 lines, and it's super cool. And you start doing yours, and I think everyone studying bioinformatics with a little bit of time in the end can do a neural network or two with a very simple code. But then once that you want to create something that is more complex, or you want to go deeper in the code and change some things that you cannot change with Carreras, then you would need to work with TensorFlow. And at least in my experience, like at least in 2019, 2020, TensorFlow was a pain in the ass to work with. Like you end up with an error log that is, I don't know, hundreds of lines long, impossible to understand. No one had the same problem in the internet. That was my experience, of course. I also learned everything about machine learning during this project, not an expert myself, but as I said, if I could start from scratch, I think I would try to start with PyTorch. I think in the long run, it's going to give you more satisfaction, and you're going to encounter less problems, less errors.
And there is any kind of tutorial or any kind of web page that you recommend to start learning about deep learning and everything about
Carreras, the page of Carreras have a very cool documentation, both how to build your models from scratch, but also cases. And I think there are some of them applied to single cell genomics, but not only about convolutional neural networks, but also generative adversarial networks, transformer, language processing, natural language processing, like a lot of them. I think it's very cool and actually easy to understand. I think I would avoid TensorFlow. I don't know how is TensorFlow now. Maybe it changed a lot. But also there are very cool blogs like Medium and Data Science. What was that? 2 Earth Data Science. They have very cool posts and very good explanation, very easy to understand with the code to follow along. I think that's very nice to follow.
Okay, we'll take note of that. They ask us also if you need GPUs in order to run the algorithm, or you can run it on CPUs as well.
So you could run it just in CPUs. Of course, it's going to take longer. GPUs accelerate the whole process. But we found out that the main bottleneck in the computational time is actually MS prime. So the simulation engine that we use, because some parameters are actually fast to produce the time matrices, but some other parameters, like very, very big recombination rates, it just takes forever to simulate. So I think that's the main problem, the computational time.
Okay, another question. Have you tried a standard DNA using a neural network as the generator?
Yeah, I have tried that. I was in the beginning like, okay, why don't we use this standard one? That would work maybe. But just by creating a neural network that was a generator, actually didn't really produce very accurate genotype matrices. And also one cool thing about this approach is that you end up with the genotype matrices, but also you know the value of the parameters. Like, you know, okay, this recombination rate can explain this, or this effective size can explain this population before the migration, whatever. But if you have the neural network, then you just end up with another batch of matrices that doesn't really explain anything. You just have weights and more matrices.
Okay, thanks. I think it's going to be the last question, more about what are you actually doing now? Like, is your research related with this, or is it just completely different?
No, it's completely different, I would say. As you said, I have moved to Barcelona. And we are in the Solar System Genomics Lab. We work with single cell datasets, not only genomics, but also chromatin accessibility. And I would say right now I'm mostly analyzing data set, and I'm now working on an atlas of chromatin accessibility of the pancreas. But I would definitely like to dedicate more time to deep learning and machine learning. I'm developing one small application in order to annotate a query data set of single cell using a reference. I am trying to build it that it works both in R and Python and with different modalities, but it's still on the way.
Thank you. So I think it's all. We don't have more questions, but before ending, I would like to, if there is anyone new, wanted to speak about a little bit our association. We work, so we are BioInfoGRX from Granada, but we work with people from all over Spain, like it's my case, because I'm not from Granada. We do different activities as talks like this, different, we are doing also tutorials, we do workshops, and our main aim is to actually get to know the bioinformatics world and how it's rising, all the data analysis in the bio world. And hope you can join us to the next activities. Actually, we have another one next week, if I'm not wrong, on Thursday, the 28th, about how to keep mental health during the PhD. I think it's going to be really useful for anyone who is doing a PhD. So hope you guys join us in these activities. And that's all. Hope to see you in the next ones. Thank you.
Thank you so much.